So whoever is there, just everyone introduce yourself, just one line, and then I'm going into the class. So there's Manikant. Please introduce.
Hello, sir. Good evening. Sir, myself Manikant Kashyap from Bihar in India. And sir, recently I am moving to class 11th and I am a NEET aspirant, sir.
Anyone, just keep on saying. We need to make the class very interactive so that we have to discuss some topic, because there are many questions in INBO or OCSC camping. We find that those questions are like open-ended questions. That means the questions are not having any exact answer, but we need to choose a very appropriate answer among the 4 options. So
Yes.
the more we discuss with the questions and answers or the topics, we'll be able to find out the appropriate answer from the options, because it's not like direct NCERT. The answer is direct from that line or that stranger. So usually it doesn't happen like this in at least national Olympiads.
Good evening, sir. I am Anis Mandal from West Bengal.
Sir, myself Ishan Banerjee from West Bengal, Bankura, sir.
Good evening, sir. My name is Adhar and I am from Uttar Pradesh.
Okay. Ankit?
Good evening, sir. Myself Ankit Kumar from Jharkhand, Ranchi.
Okay. So before going into say the chapters, say cell, biomolecules, and all these important chapters of NSEB, first we'll go through some concepts which are related to a little bit of physics and how these are associated with biology. So first, the concept we need to understand is bioenergetics. But bioenergetics is a very, very big branch. Bioenergetics. So it's a very big branch. But first, we need to understand how this concept of energy is constantly linked with the cells. So as I have already said that I'll be using that book, Life, the Science of Biology. So I have taken some diagrams and photos from that book, Life, the Science of Biology. So first, see this simple diagram. You can understand that even when a dam is the simple concept of dam, the water, the movement of water in the river, it is like associated with kinetic energy. I'll come to this, how it is connected to cell. But first, let's see this part. So the movement of water, obviously, it is generating kinetic energy. And then when a dam has been created, it stops the movement. So now the whole of the kinetic energy, which is due to the flow of water, is now converting into potential energy. So as the dam is impeding the flow of the water, the kinetic energy of water is now transferring into potential energy. And whenever we will open some section from that dam, the movement of water released from that dam is now converted into electric energy with the help of a generator. So because I don't know, this diagram is not that much clear too, but I think you got the idea. Like So first movement of water, kinetic energy, then with the help of a dam, we are just converting that kinetic energy into potential energy. And then when we again open a section of the dam and let water flow through it, so again the kinetic energy of water is now changing into electric energy. Now electric energy can be stored like in battery, or we can use it to do some work, some mechanical work. So similarly, when we see a cell, so in a cell, what happens? Any molecule that enters into the cell we need to find a way to transfer energy from the bonds of that molecule. So we need to find a way to transfer energy from the chemical bonds of that molecule. So there should be some way. And this energy can be directly used by the cell, or it can be stored. Whenever it is stored, we find a molecule named ATP. So adenosine triphosphate. I'll come to the structure and every details of all these things, but now it's just a basic idea like how constant the law of thermodynamics are applicable to a cell. So suppose this molecule is a glucose, say 6H12O6. So glucose entering, and we are extracting the energy from the glucose in some way, glycolysis, Krebs cycle, whatever the process is, we'll see that. And the energy can be directly used, or it can be stored. So this law of thermodynamics, so there is 3 laws first, but we need to mainly focus on first 2 laws. So first law and second law. Although this is a topic of physics, but we are just scanning through the fundamental nature of the laws. So first law is what? That energy can neither be created or nor be destroyed. Energy can only be transferred from one state to another. So energy can only be converted from one state to another. Sorry, then what is energy? Simply, physicists define energy by a simple definition, that the capacity to do work. Just energy is defined by the capacity to do work. Now, in case of molecules, we'll see the definition of energy changes to this chemical. In case of chemical energy, the amount of energy that can be used to do work is called free energy. So I'll come to that. So first, let's see that first we have seen that energy is basically capacity to do work, According to first law of thermodynamics, energy can only be created from only be converted from one state to another. It can neither be created nor destroyed. And in second law, the main statement of second law can be said like this: no work or no conversion is 100% efficient. So when one form of energy is converted to another form of energy, so in never ever a single conversion has been 100% efficient, that all the chemical energy can be converted into some form. So it never happened. It never happens. So some form of energy or some energy is lost as heat. And the amount of energy that is lost, it always increases the disorderness of the system. So this energy always increases the disorderliness of the system. So any system, its disorderness will increase. So now in chemistry, there is a term associated with this disorderliness, and the term is called, so the measurement of disorderness, this is called entropy. Entropy. So entropy is a term that comes from the second law of thermodynamics. So we will be dealing with entropy only. That means according to second law, this disorderness of the universe or of a system always increases, and this is natural. So what? So entropy is, you'll find all this explanation in your chemistry class, but I'm just scanning through the basics, through that we can directly come into biology. But before going to the function, how cell work or how a physiological system works, we need to understand little bit basics, like why this concept is necessary, or energy or entropy or whatever. So entropy of a system always increases. There are lot of theory with entropy. Like entropy is the only parameter that tells us about the direction of time, because along with time, always any ordered structure becomes disordered. So anything ordered, suppose these are some molecules which are like in a, say, ordered state. So as time passes, this ordered state will be disturbed, and this is like by a law, according to a law, second law. And second law is like, this law of thermodynamics is like more fundamental law than the laws of even Newton. So these laws are like more fundamental, and even all the science, the physics, chemistry, everything depends upon the validity of those laws, some basic laws. Now, what we can understand is that for any system, the entropy or the disorderness will always increase, and this is spontaneous. So for any, suppose for any spontaneous reaction, for any system, for any system, entropy is increasing. Now, suppose a compound, or say a molecule, A is converting into B inside a cell. So suppose this is a chemical reaction, A is getting converted into B. In this reaction, the amount of energy available in A for work, so the amount of energy after 15 minutes, I'll give everyone some time to ask questions, whatever we are studying now. Okay. So the amount of energy available in A to do work, or in this case, to participate in this chemical reaction or proceeding of the reaction, is mentioned by a term called Gibbs free energy. Gibbs free energy, G. So for any reaction, for any natural reaction, Gibbs free energy always decreases, because the amount of energy the reactant has, it should be more than the amount of energy in the product. So this is reactant A and this is product. So for any chemical reaction, any reactions which are occurring spontaneously, we will find that the reactant, the energy associated with reactant is always more, the free energy associated with reactant is always more than the free energy associated with product. So this can also be explained by the concept of potential energy, by the concept of potential energy. So in many books, you'll find that what happens, in many books, you'll find say this chemical A has been compared to an object. Say A is an object and this is ground. This is ground. So if we drop A, say A is a ball now. So ball, so we are comparing it to an object. So suppose a ball is released. Now what will happen? The ball should fall to the ground due to the change in. So now it is due to gravitation, but we can call it the potential energy of ball at this position is higher than the potential energy of the ball in this position. And this potential energy is actually, as it is due to gravitation, so we'll call it gravitational potential energy. So normally or naturally what happens, any object any object tends to or tries to decrease its potential energy, or more or less the potential energy is, the more we can call it stable the object is. That means when there is high potential energy, it is like unstable, and when there is very low potential energy, then it is stable. So to increase stability, any object tries to decrease its potential energy. Now here, when we are comparing it to the force of gravitation, this potential energy is gravitational potential energy. When it is due to charge, then that is electric potential energy. So the force can be different, but everything can be explained by the concept of potential energy. So even in chemical reaction, what happens? As the reaction occurred, so as this reaction occurred, that means the reactant tried to decrease its potential energy. But in this case, we are saying it with a new parameter, or those who have studied class 11, they know, like, this is called Gibbs free energy. And there are so many equations and everything in this energy, but we are not going into chemistry. So Gibbs free energy for any spontaneous reaction will always decrease. And when this energy is decreasing, so for any spontaneous reaction, G value decreases. And when G decreases, there is some amount of energy released in this reaction. And the released energy causes increase in entropy, disorderness, increase in entropy. So till now, what we have studied? We have studied, like, for any cell, there should be constant supply of energy. And this supply of energy is occurred from surrounding. So the surrounding of any cell is very important. So from the surrounding of a cell, what's happening? Some molecules enters or diffuses into the cell. And from that molecule, cell tries to extract energy. This energy can be directly used by the cell, or it can be stored in the form of a chemical bond. Now, what is energy? Energy is the capacity to do work. And even cell follows law of thermodynamics. First law is just energy cannot be destroyed, cannot be created. Energy can only be converted from one state to another. Second law is no conversion is 100% efficient, so some energy is released, and that energy always increases the disorderness of the system, and we call it entropy. And so a very short version of second law is entropy of a system always increases. Now, as so in a system, we have seen, like, what happens in case of chemical reactions that are occurring inside cell. So say in a reaction, A is converting into B, so some energy is getting released, because in any spontaneous reaction, free energy value always decreases. And free energy value is explained by G. And it can also be explained with the example of mechanical energy, concept of mechanical energy. So there are two forms of mechanical energy, potential energy and kinetic energy. So it can also be explained with the example of potential energy. Okay, so if there is no question, we can now just check this reaction. This is also from the a book like The Science of Biology, or even you'll find this in what Campbell, same. Tushar is asking if we convert any energy into heat, then 100% efficiency. 100% efficiency, I'm saying in terms of work. If you can use the total amount of heat generated by you to do some work, then yes, it is 100%. But if you cannot do any work with that energy, then the conversion is not 100%. So why we are trying to convert some form into another form? Because so that we can do some work with that energy. But usually what happens, any conversion, the amount of energy that is lost as heat, it is not used in to do some work. It is not used to do some work. So that's the answer. Kishan is saying how released energy causes increase in entropy. Okay, I don't know how, means in what sense you have asked, but in a sense this question is also very deep kind of question, but I can answer it like vaguely because it is not required that much. Suppose in any reaction when heat is released, heat actually what it does, it actually increases molecular vibration. So when there are a lot of molecules in any area, so all the vibration of the molecules increases with the generation of heat. So as there are more vibration, there can be more scattering of the molecules. So as the heat is simple example is like when the environment is more warm, you can smell things like from a real distance from yourself. So say there is a food, so you will be able to smell the particles, because the particles that are released from the food are having more vibration due to warm environment in comparison to very cold environment, because then the vibration will be less and the diffusion of the molecules will also be less. So this is like a simple real life example when we can understand that the more the heat is, the more scattering of the molecule is of the molecules here. So the more scattering is, we can say the more disorderness is there, and the measurement of disorderness is entropy. Okay, now if we can go through this kind of reaction, we can understand that the ATP, so I've said like ATP is the is like a small battery in our cells, where I've said this. So energy, if it is stored for any future use, suppose
Will
like
you please tell me how the ATP is formed?
oh, okay, so let's see what is ATP. See, this molecule ATP, or whatever we call it, adenosine triphosphate.
How the ATP is formed? मतलब I think ADP plus PI is equals to ATP. No, that I am asking.
Yeah, I'm just going through that. See, to form this molecule, say adenosine and then phosphate and then 3-phosphate, first there should be a sugar. So cell uses Like when we are having glucose, we are not storing it for any purpose. We are just breaking the whole glucose to get energy from it. But there are some sugars which we are not breaking. We are keeping it so that we can make it a battery. So 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 adenosine triphosphate can be compared to a small or microscopic battery inside cell. Now this battery is composed of 3 units: sugar, nitrogenous base, and phosphoric acid. These are the 3 compounds that is needed to make this smaller battery. Now, this sugar is ribose sugar, which is also found in RNA. Ribose. And the nitrogenous base, this is also a chemical compound, a 2-ring structure, which is called purine. Just purine, but exact name is adenine. So when ribose sugar is added to adenine, we call it adenosine. And the phosphoric acid, H3PO4, when phosphoric acid is attached to adenosine, we'll call it adenosine monophosphate. Adenosine monophosphate, or AMP. So the structure looks almost like this. So this is the sugar. This is first, second, third, fourth carbon. In the first carbon, there is attachment of a nitrogenous base. We call it adenine. And in the 5th carbon, there is attachment of phosphate. P double bond O. This is the phosphate attached. So this is the first. This is the structure of adenosine monophosphate. But this structure is more stable. Stable structure. But we need to form an unstable structure, because stable structure will not break down to give you energy. Any structure which will give you energy should be a little bit unstable. Otherwise, it will not break itself to give you energy. So what happens? Now, if this AMP, this structure, now see H2, everything is same, just I'm changing here. This phosphate is binding to now another phosphate. Now this is becoming what? ADP, slightly unstable than AMP. So when we are breaking down glucose, the extra energy we want to store, we can store it with the help of extra phosphate. So to get this phosphate, we are using what? Our cells are using phosphoric acid. So they are getting this phosphate from phosphoric acid. Now this ADP, which is slightly unstable because of the phosphate-phosphate bond. This bond is very unstable. So, and then the cell is using, there are some questions, how this structure is unstable. So I'm saying like this bond is like more unstable when you attach one phosphate with another phosphate with the help of phosphoester kind of linkage. This is a real, this part is like more unstable. Now, instability and stability is determined by resonance formation, or how to stabilize these charges. But this is like more chemistry, so I shouldn't waste more time just to describe the bonding nature. I'm just trying to answer the question of how ATP is the energy currency. So similarly, one more phosphate will be added and it will become ATP. And ATP has very less half-life. So anytime it can break and it can generate ADP again and phosphate. And if you wait for much more time, sometimes even ADP can be broken down to generate phosphate. PI means inorganic phosphate, to generate phosphate and to release AMP, which is like very stable structure. So in every step, not in every step, but in this step, there is a lot of energy released, which is 30.5 kilojoule. So Ankit Kumar is asking, what is the difference between ATP and DATP? Okay, good question, but here actually D and L form of ATP, so it mainly depends on the sugar. So it will become very complicated now if I try to explain sugar, because I was having what should I teach, like after completing, say, until metabolism, I'll come back to this part again if we have time today. So you know, this amount of energy is available to cell for some other reactions, or for some, say, what to say, for some mechanical work to do. So whenever ATP is so ATP is a very unstable compound. It is generated in few processes. There are very few reactions where ATPs are generated. Whenever it is generated, it can just go to another reaction and release its energy. So the example is here. You can see. Sorry, it is in calorie, not kilocalorie. It's in joule maybe. 30.5. So now you can see this reaction. In this reaction, basically, whenever ATP reacts with water, it forms ADP and phosphate. But the amount of energy released in this reaction should be utilized as soon as possible by another reaction. So here you can see the reaction is glutamate, some other compound, glutamic acid, is reacting with ammonium ion to form another compound called glutamine. So there are thousands of reactions like this. This is just one example. But what is happening? As soon as ATP is broken down, the energy released from one reaction should be utilized by another reaction. Otherwise, the whole process is waste. So ATP breakdown, or we can say hydrolysis, ATP breakdown or hydrolysis is always a coupled reaction. That means it should occur simultaneously with another reaction. Just occurring of ATP breakdown makes no sense in a cell. Fine. So now let's recap what we have learned till now and make some sense out of it. We have seen that energy flow of energy is vital for survival of a cell. Now this energy flow usually occurs in the form of a chemical. So the form is actually chemical energy. And the chemical is known as food. So any chemical from which we can extract energy, that chemical is actually food. So glucose is a food for us. Glucose is a food for cell. So glucose is a food for the cell. And what glucose does, it is actually carrying some energy within itself. Now cell have to extract that energy from it. So in any reaction, we know that the free energy associated, or sorry, available to glucose, so we are calling it G. Not for glucose, just for the free energy from Gibbs free energy. Now someone said, like Manikant is saying, sir, is this process is known as Krebs cycle? Because in 10th class I have studied the ATP is formed by Krebs cycle. Now Krebs cycle or glycolysis, all these steps are just breakdown of glucose. How we are breaking down glucose. Now if we start from glucose, say C6H12O6, there are so many steps, and at the end of those steps we are left with 6CO2, and there are some H2O, 6 CO2 and 6H2O. So we'll be having 12 and 6 and 6. Okay, I'm not going to because this is not a single step reaction, so you cannot actually balance the reaction, because these are like, there are so many things going in and out. So my main focus is like how the whole glucose is now converted into CO2, and CO2 we are releasing from our lungs. So breathe out from the body. So the glucose is completely converted into CO2 after two steps. One step is glycolysis, another step is Krebs cycle. So after these two series of reaction, glycolysis is one series of reaction, Krebs cycle is another series of reaction. After these two series of reaction, a complete glucose molecule is left with nothing but CO2. But in all these steps, in so many steps, there are release of energy. And cell tries to trap this energy in the form of ATP. So there are so many ATPs formed during this conversion of glucose into CO2. So the free energy available to glucose, say we are denoting it by the term G. Now, say glucose and to CO2, say this is G1 and say this is G2. G2 should be very, very, very less than G1 because the amount of energy released, we have already converted it into our energy currency. So as we have already transformed this form of energy to ATP, so G2 should be very, very less than G1. Now, if we try to find out the difference between these two Gibbs free energy, so we call it del G. Del G, difference between the Gibbs free energy associated with the chemicals. Now this del G, what happens? Del G is actually products del G minus reactants del G. Product minus reactant. And as product is so low, so if we subtract a large number from a small number, what should happen? It should be a negative number because this is small, G1 is large. So del G is negative. This is what you have observed here. Del G is minus 7.3. So the negative sign, whenever you are seeing this negative sign in del G, that means the energy associated with product is less than the energy associated with reactant, and it should happen because this is what natural process is, right? This is what we have seen, like for any spontaneous reaction, G value always decreases. Then this should be the natural reaction. What is unnatural, what shouldn't happen, or what is like beyond understanding regarding the theory we have learned till now, that endergonic reaction. Usually in this reaction, if you see the reaction, you'll find that if you don't say I'm just working on something like this. Suppose you don't know the upper part. I'm just blocking it. You don't know the upper part. In this case, if you just see the reaction below, you'll find it confusing that it is not this reaction is not following the second law, because what is happening here? In this reaction, you can find that the del G, that means G of glutamine is more than the G of glutamate. That's why the number is positive. But positive number shouldn't happen, because we have seen that this is a general idea that for any reaction to occur, del G should, G should decrease. But here, how G then increased? So then you have to think, okay, so there must be some form of energy which is supplying in this reaction. Some other reaction should be there, which is actually giving or supplying this energy to the reaction. Fine. So I think you have got the idea that there are two different type of reaction. One is exergonic, which is normal and which should happen according to thermodynamics laws. And another is endergonic, which is in which for the particular reaction del G is increasing. But if you see the combination of two reactions, so as you can see, in one reaction there is negative 7.3, and in another reaction there is positive 3.4. If you total this, you'll find the overall change is what? Positive or negative? The overall change is always negative. So one positive, one negative, but the overall change is always negative. And this is how even these reactions are following the second law of thermodynamics, that ultimately del G value should be negative. Although in some reaction, you'll find that there are positive del G. Okay, so there is some question. Anish is asking why the difference between G1 and G2 is very big. Oh yeah, G2. Because from glucose has so many chemical bonds, and when you are breaking down all these bonds, you are releasing a lot of energy. If you break down just one bond, some amount of energy will be released. Another bond, so there are 6 carbons, so many bonds. So as you have broken down so many bonds, and ultimately you are left with just carbon dioxide, so there must be so much of energy which has been wasted or lost from the structure. That's why the difference is more. Okay, asking, sir, can we call this type of reaction as endothermic, exothermic? Yes, very good. So endothermic and exothermic reactions are also this type. So same, just terminology is different. So exergonic, we can call it exothermic, and endergonic, we can call it endothermic. Okay, so we are just summarizing that energy flow occurs in the form of a food, and the food, say glucose is a food for cell, glyceraldehyde is a food for cell. So for any reaction to occur, the free energy always free energy of a reaction or of a process, whole process, process means what? Process means actually series of reactions, like glycolysis. There are a lot of reaction. Krebs cycle, there are a lot of reactions. So when the free energy of a process means of a series of reaction also tends to decrease following the law of thermodynamics. Now, after all this energy flow, finally, we can understand that some energy is stored in the form of ATP. Not only ATP, even GTP. ATP is the major. Like you can say ATP is like dollar of this currency market.
Sir, what is GTP?
GTP means guanosine triphosphate. There are CTP as well, cytidine triphosphate.
Sir, can you obtain energy from GTP and CTP
Yes,
that
yes.
can be utilized by cell?
Yes. So there are many reactions. When we'll study respiration and photosynthesis, we'll find there are some reactions where we can even extract energy from GTP. So ATP is the major currency. So most of the time we are using ATP. But GTP, CTP, there are some few other high energy compounds as well, where which cell can use to study. Cell can use to run its say mechanical work, osmosis work, whatever kind of work. So this diagram is also very important from the book, and we have seen like this type of questions coming again and again in Indian National Biology Olympiad. So exorganic reaction can be explained with the help of a ball falling from a say hill type area. So if you release this ball, from a height, so obviously the ball will fall down. So this is like more obvious kind of reaction. Every reaction should be like this. And in case of endergonic reaction, what it seems like that you have released a ball at the base of a hill and the ball is climbing up. So this is not obvious. This is like unnatural kind of reaction, but we know how this occurs from the last example. Okay. So endergonic, exergonic, we are very clear after all these examples. We are also very clear that free energy decreases, and we are clear that after those due to this decrease, some form of energy is stored in either ATP, GTP, or CTP, or any other high energy molecules. We'll find creatine in phosphate in muscle cell. Okay, so our point was living any object or anything that is living, should be able to revert or I can say maintain orderliness in the structure or in its structure. Now the orderliness is the internal orderliness. So any cell or any living organism, any living object should be able to defy the second law of thermodynamics. Because suppose this is a cell, and if we explain it like this, suppose these are some molecules forming the boundary of the cell, and all these molecules are in a very ordered state. Now if due to some, and we know from the second law that this orderliness shouldn't be there for a long time, because with time this orderliness just will be destroyed due to this law. So cells should find some way so that it can maintain the structure. So any living organism or any living object is those objects which are or which can maintain the structure and defy the change of entropy. And this concept is also present in the CBSE class 10 life processes chapter, first page. So living things should be able to maintain their structure in spite of being the structure getting disordered due to that or due to a physical law which is applicable for any object on earth. So with this we have understood a little bit of basics of this of the cellular organization and living organism. Now, a very prominent question that comes in biology, that is, why cell is small? Why cell is small? So there's like a very naive question. You'll find that why cell is small means what? Cell is small, so small. But this is also a very fundamental question. Why cell is small, or why cell is microscopic? Why we need microscope to see a cell? Why a cell should also a cell can be a large big ball you can find or you can say through your naked eye. Why cell is not like that? Why we cannot see a cell through our naked eyes? Now, can anyone just answer anything regarding that? I'm asking, can we call this to, oh, maybe I have answered this.
Sir, maybe because the atoms constituting the cell is microscopic.
No, my question basically is like this. Suppose all the macroscopic object we are seeing, suppose I have a grape here. Suppose I'm just bringing a grape. So this is a grape, right? So this is a fruit. And now in this grape, there are millions of cells. Why it is not like that, that this whole grape can be a single cell so that I can see it by naked eyes? Why multicellularity arise? Why there are what is the need of multicellular organism? It can be like a big organism, but the whole organism is a single cell. Why something like this cannot happen?
Sir, maybe the reason is that because an organism needs a various work, example, excretion, digestion, and etc. And if the cell will be single cell, then it can't be work on a very great manner. Example, multicellular can do, because when one type of cell will group, that will tissue will be different from another, and the work will be um, defined.
Yeah, means idea is almost correct. But we will try to explain it with the help of some physics.
Sir, there can be division of labors.
Yeah, but if there is single cell, there is no there is no need of division of labor. So why we need division of labor? Single cell could have done the work.
For emergent properties.
Emergent properties. Okay, so that is a emergent properties is very different, but there is also a view through which we can answer, but still it is not answering one fundamental question, like what is the necessity that even for emergent properties too, because first it has to it has to come out of the unicellular nature. It has to come out of unicellular nature. So suppose I am saying that okay, actually this is a very basic question in Campbell, in any other uh, international books you'll find. Suppose this is a cell and this is a this is having a cubical structure, cube. Now say each side of the cell is 1 millimeter. First you need to understand that the surface area, so the surface area of a cell yes, very good. So ratio of cell surface area and volume. This is actually the most basic answer, most fundamental answer. So the surface area is what? Surface area is required to collect molecules from environment, because the more the surface area is, more molecules it can collect from the environment. Collect molecules from its surrounding, or from, yes, from surrounding. And also surface area is needed to release the waste product that is being formed inside the cell. So surface area is needed, high surface area is needed to collect molecules from surrounding and release waste to the surrounding. And what is volume is doing? What volume is doing in this case? More volume means more reactions should occur. I don't know you're understanding or not. Surface area, what is surface area? What is the role of surface area? Sorry, role of surface area. So the more the surface area is in a cell, the cell can collect more molecules from the surrounding and release more waste to the surrounding. So cell will be more efficient. What is the role of volume? Now, role is like volume provides the area where reactions can occur. All the cellular reactions, which we call metabolic reactions, all these things can occur. Now, if you know, there are say if the volume increases, cell should perform more reactions, because to maintain the whole volume they need more materials now. And how will they obtain more material? With the help of their surface, which is in contact with the environment. So the surface is in contact with environment. Now as volume is increasing, cell needs more and more reactions to occur, because they need more products to maintain this whole surface, whole volume. So more reactions are required to generate more products So more reactions are required to generate more products so that a cell can maintain its internal environment. So that a cell can maintain internal environment. So to maintain the environment, maintain the internal environment, they need more and more reactions to occur and more products to form. Now let us see. When a cell is having very less, say the size of this one side of this cube is 1 millimeter, then the area is the formula is 6a square. So area becomes 6 into 1 square. So 6 square millimeter of this cube, and the volume of this cube is a cube. So a is 1 millimeter. So 1 cubic millimeter. So what is the surface area is to volume ratio? It's 6:1. Now we need to understand that a cell needs to maintain a very high surface area with respect to its volume, because they need more and more flow of molecules from outside and also flow of waste from inside. So a cell needs to maintain high surface area is to volume ratio. Ratio should be high, like 6:1 is still good. Now my question, why cell is small? The less the size is, the more surface area is to volume ratio is. So less size is equivalent to more surface area is to volume ratio, and more surface area is to volume ratio is equal to more activeness or more efficient cell. Very important. Cell is very efficient. Why? Because now they can easily transfer waste products outside, easily acquire raw materials from outside, because surface area is more in comparison to the volume it has. And what if surface area increases slightly? Suppose if you increase 1 millimeter to 2 millimeter, so this cube is 2 millimeter. Now what will happen? Area becomes again 6a square, so 6 into 4 square. Sorry, 2 square, sorry. Means 6 into 4. Two square. So 24 square millimeter. And volume becomes how much? 8. Now what is the area is to volume ratio now? 3:1. You can see there were 6:1 when 1 millimeter size. Now with just 2 millimeter size, area is to volume decreases significantly. So significant decrease in area is to volume. So cell will now suffer. Means as so you all can easily understand, as the cell size is increasing, so we wanted to see cell through our naked eye. So now as the cell size is increasing, what is happening? Cell itself is not able to survive in this condition, because they are not getting enough surface area for the amount of volume they are having. Similarly, you'll find if the size is more, say 3 millimeter. Now area 6 into 3 square 9, volume 3 cube. How much is that? 2:1. Very less. This inside volume cannot even, the amount of diffusion needed to maintain all this volume inside cannot be compensated by the surface area they are having. So very less area is to volume to maintain internal environment, or yeah, we have seen internal environment. So I think you have understood. So as a cell tries to increase its size, there are many cells which are like very big, like a neuron. Neuron is so big that even there is some neuron like from your vertebral column, say to the tip of your finger. So this is a long neuron. So how such neurons can survive? So what happens? So what happens for a large cell to survive, for a large or say long cell to survive, it needs to increase its surface area by other means. Needs to increase its surface area by other means. Other means like, what does a neuron perform? You can see, suppose if this is a cell, and if it's just elongated, it's very long, it would be very tough to survive like this. So what a cell does, neuron, star shaped, so it tries to make so many processes so this pointing structures are called processes of neuron. Why they are making this type of structure? Even to survive itself, to increase surface area, more and more surface. So any large cell which tries to maintain its say length, or if a cell is very large or very long, those type of cells tries to compensate their volume with the help of some kind of processes coming out of the cell, so that the surface area of the cell remains very high. So okay, Anish is asking which ratio is perfect. Perfect, I don't know how to say perfect, but yeah, for good surface area to ratio is around 8:1, 8, say 8:1 to 10:1, some kind of this type of ratio. So, okay. So this is the answer of the question, like why cell should be small, because cell have to survive. Simple. So just to survive itself, it has to be small. It cannot be big. And if it's big, then it will make some branch or something like this so that its surface area is increased. So this is another basic concept in biology. And although we have seen all these reactions, but metabolism, we need to understand metabolism. What is metabolism? Metabolism is the sum total of all the chemical reactions that are occurring in a living body or in a living cell. So metabolism is the sum total of all the chemical reactions that are occurring inside of a cell or inside of a living body. This now any reaction, say A to B, in other parts say C to D, say X to Y, all these reactions are metabolic. Collectively they are called metabolic reactions. Now now similarly, if the reaction is breakdown, now whenever we are thinking about breakdown, something should strike like exergonic, because entropy is like more disorderedness. So when something will break down, the things will be more disordered, obviously. So whenever suppose A is breaking down into B and C, that means ki so a molecule A is broken down into B and C. So a lot of energy is released because bond is broken down. So breaking of bond, breaking of a chemical bond is what a lot of energy is released. So when a lot of energy is released, so the Gibbs free energy G of B and C should be, so G of B or G of C or plus, say G of Gibbs free energy of B and Gibbs free energy of C is still very less than Gibbs free energy of A, because a lot of energy has been released because the bond has been broken. So in such reactions, so obviously del G is negative. And energy is released. This is exergonic reaction. But this is, in biology, we also call it catabolic reaction. This type of reaction, whenever there is a breakdown of chemical bond, this type of reaction is also called catabolic reaction. And most of the majority of the reactions inside our cell is a catabolic reaction. So catabolic reaction is a type of metabolic reaction. And similarly, the things we have seen last time, this energy, if it is used to create bond, say P plus Q. So these are two different compounds, but they join together to form a compound R. But it is not possible normally. But in cell it is possible because cell can store energy in a form of ATP, and then this ATP can transfer energy to another reaction. Now in this reaction, there is a formation of chemical bond. So to form a new bond, you need energy. And now this type of reaction in R gives free energy of R is obviously more than the gives free energy of P and gives free energy of Q, because we have extra supply of energy. So obviously del G becomes positive here. So this type of reaction is endergonic. But in biologically, we also call it anabolic. Anabolic reaction. So metabolic reactions can be classified into anabolic and catabolic reactions. Now usually whenever our body tries to make something, suppose amino acid और amino acid के बीच में bond form करना, so this is kind of anabolic reaction. So suppose one amino acid and another amino acid reacts, or say many, a lot of amino acid is reacting ... to make what? Protein. So every time a new bond is forming, new chemical bond is forming, this reaction, obviously synthesis of protein, this is a anabolic reaction, and it requires a lot of ATP. So to make protein inside our cell needs lot of energy supply. But the question, the equation we have seen regarding glucose metabolism. So when glucose comes into the cell performs glycolysis, and then it performs Krebs cycle, and then finally from each carbon of the glucose is now a carbon dioxide. So there are 6 carbon in a glucose, so there should be 6 carbon dioxide. I just think, can you repeat this? Which one should I repeat? Which one to repeat? Last part means protein, amino acid protein, or glucose. Okay, amino acid protein. So amino acid protein, what is protein? Protein is a structural protein helps in forming the structure of cell. So any structure in living organism, any kind of cell, whether it may be amoeba, paramecium, or human cell, RBC. So the structure of the cell is very much dependent on protein. So this is just one idea. Now, when one amino acid, so to make a protein, there should be a lot of amino acids, even thousands of amino acids. So amino acid and amino acid are reacting together to form new bond between them. and to make new bond, we know that for the formation of any chemical bond, we need supply of energy. So we need a lot of energy supply, and it will be provided by ATP. The question asked by Ankit is, can we make polymer by removing water, just like hydrolytic enzymes break polymer by adding water? Oh, yeah. So can we make polymer? Oh, yes. Uh, I couldn't understand at the first instance what you were saying. Yes, we can make polymer by removing water. That's why this type of reaction is called condensation reaction, in which a water is released. Okay, very good. Manikant is asking, sir, when G will be positive, then the reactions will be anabolic, or there is any exception? There can be a lot of exceptions, because no anabolic or catabolic reaction is a single step reaction. So what happens, like there are series of reactions, so 1 2 3 4, say there are 10 reactions. So you can find, like, even in a catabolic process, you can find, suppose what will happen, like in a catabolic process, majority of the reactions, say 90% of the reactions will be what? Del G negative. But there can be, in between, there can be 1 or 2 reactions where you may find Del G is positive. So in between, say there are 10 series of reaction, and in between 4th and 5th reaction is Del G positive, but all other reactions are negative, and in general total is negative. So in case of anabolic reactions, if it is a single reaction, then yes, always anabolic reaction is positive and catabolic reaction is negative, if it is a single reaction. And if it is a series of reaction, then there can be exceptions. Okay, so if there is any case that G becomes 0, G becomes 0. G becomes 0, then there is no reaction, obviously. So there will be no reaction. Any reaction, there should be change in free energy. Okay. So we've seen a little bit idea of metabolism, and the other details of glycolysis, Krebs cycle, we'll see in other in the actual chapters. And we have an idea of this part. We're fine. Okay, so protein. Okay. Now, before going to cell, we need to understand at least one part of the biomolecule chapter of class 11 NCERT, and that is protein. Protein is like very, very important to understand any other chapter, or any other chapter fundamentally or in details. So we need to understand protein, and this will be like a topic of biomolecule. So we are now going into more detailed chapters. Before that, you can also play with something called Fold It. So there is a website, just checking it. Wait. I think this should be there. Yes. So there is a website. Called fold.it. When we will study protein, you guys can go to this website and play with the structure of proteins. So this is a there are 4 US universities who came up with this idea, and students can play with the structure of protein. And even some students have discovered some new structure of protein after playing in this website. So you guys can check it later after I complete this protein part. So protein is a polymer composed of amino acid. Protein is a heteropolymer. What is heteropolymer? So if a polymer is composed of dissimilar types of monomer, so if a polymer is composed of dissimilar types of monomer, then this type of polymer is called heteropolymer. Dissimilar, so there is differences in the monomer. Like when we understand starch is made up of glucose. So glucose. So if there are thousands of glucose and all glucose are same, it looks same, structure same, so they are not, starch is not heteropolymer then. Now if any, like DNA, in a structure of DNA we find A, T, G, C. So each monomer is different from another monomer. So whenever any polymer is composed of dissimilar types of monomer, they are called heteropolymer. So protein is a heteropolymer because protein has 20 different types of amino acid. So protein has 20 different types of amino acids. Now when a structure is having 20 different types of amino acid, it should be a hetero. But so this is a very tricky line, tricky statement, that protein can be homopolymer too. So you can also make homopolymer through means you can also make a protein a homopolymer. How? If a single type is repeated 1,000 times to make that protein. So if a single type of amino acid is repeated numerous times, several times, to make that protein, obviously. So to make that protein, if you use one type out of that 20 types, then that protein can be homopolymer. But naturally, it is never found. Naturally, always proteins are heteropolymers. Now, before going to proteins, Ankit is asking what is 1 degree, 2 degree, 3 degree. Yeah, these are very simple things in class 11. So very easy things, 1 degree, 2 degree. But before before coming to the proteins, I need to first explain amino acids. And it's very, very important, amino acid. So I have 3, 4, 5 minutes. So just 5 more minutes because I have another class. So I'm just looking at a structure of amino acid. What is amino acid? First we just see the chemical structure of amino acid. So the structure is very simple. There is a carbon, and in place of hydrogen, in one side there is COOH, another side there is NH2, one side there is hydrogen, and there is an alkyl group. Alkyl group means you can add any number of carbons here or any structure here, hydrocarbon. So this is the acid, carboxylic acid functional group. And this is NH2. I have written it in a reverse way because the bond is with nitrogen, that's why. So NH2, this is actually what? Amine group. Amine group. So a structure having amine group and carboxylic acid group, and there is alkyl group. This structure is called amino acid. Now here carboxylic acid is the main functional group. So you know in organic chemistry you need to find out which is the priority functional group. So carboxylic acid is the main functional group here in a amino acid. That's why acid is the last term. Otherwise we would have called it amine, something amine. So now the main functional group attached
Sir?
to the yes.
Sir, why can't we substitute alkynyl or alkynyl groups in place of R?
Yeah, we can substitute. We can add anything here. You can add say CH, say CH to CH3, or you can add say CH double bond CH2, or you can add something triple bond, anything. So this group is overall an alkyl group. You can add alkene, alkyne, or anything. Okay, so this amino acid structure, the carbon that is attached to the main functional group is called alpha carbon. So the carbon attached to the main functional group is alpha carbon. And amine group is attached to alpha carbon. That's why this amino acid is also called alpha amino acid, because the amine group is attached to alpha carbon. And what is alpha carbon? Alpha carbon is the carbon adjacent to the main functional group carbon. So you can ask then what different kind of carbon can what different kind of amino acid can be there. So if I draw an amino acid like this, suppose C, 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 and now here there is NH2. So and suppose there is long R group, there is H group. Now the main functional group is COOH, and the carbon adjacent to the COOH is alpha carbon, and the this carbon is beta carbon, more away from COOH. And the third one is gamma carbon. Now the amine group is attached to gamma carbon. Now we can call it gamma amino acid. You just add HH. You know, this amino acid never forms protein. Only alpha amino acid forms protein. So the kind of amino acids that are found in protein, those are only alpha amino acid. Beta, delta, or gamma, all those other amino acids never forms protein. But there are some other roles. Like this gamma amino acid has a very important role. This amino acid is also called GABA. GABA. What is GABA? This is called gamma amino. How many carbon are there? 1, 2, 3, 4. Let me remove this R with H. If I just remove this R with H, then the whole structure becomes, there are 4 carbons, so butyric acid, and in this butyric acid there is a amine group attached to which carbon? Gamma carbon. So you can call it gamma amino butyric acid. So this gamma amino butyric acid is so important that it is also found in our body, and it acts as inhibitory neurotransmitter. Inhibitory neurotransmitter. So some neurons of our body releases this structure as a neurotransmitter. Suppose neurotransmitter, what is the function of neurotransmitter? Neurotransmitter means released from nerve cells. So basically, suppose हम लोग जब नींद में रहते हैं, तब the movement of body gets reduced. So otherwise, when we are awake, we are moving our body parts here and there all the time. But when we are sleeping, all the body movements are suppressed. This suppression of the movements of our body is also done by the nerves using a neurotransmitter which is inhibitory. Usually when I'm moving my fingers or something, I'm talking, the nerves that are releasing neurotransmitter, those neurotransmitters are exciting my muscles, so excitatory. But here in this case, this neurotransmitter, very important example, GABA, this neurotransmitter released from nerve cells, but these neurotransmitters causes suppression of the muscle activity. So our muscles will not move. So during sleeping, this type of neurotransmitters are released. And if these are not released, so you know, some people are having sleepwalking, some people do sleep talking, all these things. So it can, all those things can happen. Okay, I'll leave here for tonight. There's some question from Ankit, sorry. I can read full chapter Energy and Life after this lecture from Campbell. Yeah, you can read that chapter, or you can also wait, there is a chapter called yes, so there's the chapter number 6 of the book I have sent in the group. So you can see 5 and 6, chapter 5 and chapter 6. So you can read this too. It's not that big. very easy with lot of pictures.